Thank you, and yes, I'm super excited to be here today to share some work with you on improving monitoring of clonal hematopoiesis. So hematopoiesis is the process of blood production. Our hematopoietic stem cells live in our bone marrow and give rise to a number of different cell types that differentiate into all of the cell types that make up our blood. For the purposes of today's talk, I'll be just talking about three categories of cell types: lymphocytes, granulocytes, and monocytes. So in clonal hematopoiesis, the hematopoietic stem cells receive a somatic mutation that then changes the behavior of the hematopoietic stem cell. They have a proliferative advantage, and they also bias their production towards myeloid cells. And depending on the mutation that they receive, we often see that heading towards monocytes. And so when a patient has clonal hematopoiesis, you're able to detect a growing clone of mutant monocytes in their blood. So clonal hematopoiesis itself is a pre-malignant condition, but we do recognize this as a significant risk factor in progression towards blood cancer. Some work in the UK Biobank has established that depending on clone size, we see increasing risk of progression towards blood cancer. So currently, the method for monitoring clonal hematopoiesis relies on next generation sequencing, specifically DNA sequencing of peripheral blood. And so there are a number of genes that we know get mutated in this process, and so targeted sequencing looks for mutations in this specific set of genes. And basically, you count how many mutant reads you have at those locations compared to the total number of reads, and that proportion is called variant allele fraction, or VAF. So I want to just consider a case of a patient who is being monitored for clonal hematopoiesis. So let's say this patient comes to us in January and receives testing for clonal hematopoiesis. And in January, we find that they have a clone with a VAF of 0.1. So that would be 10% of their reads. And so this is a fairly large clone that we might be concerned about. So let's schedule a follow-up appointment with this patient. So in July, the patient comes back to us for their 6-month follow-up appointment, and we do this test again. And now we see that their clone has a VAF of 0.3. So it's a significantly larger VAF, and it's very large. And so this might be something that we are very concerned about. So what does this mean for our patient? Well, I'd like to consider two possibilities about what's going on for this patient in their blood. So the first possibility is the clone itself is expanding rapidly. And so if we think about the cell types that are making up their blood, let's look at these lymphocytes, granulocytes, and monocytes. So in the first case, in January, we would observe a small fraction of their monocytes are mutant. But if their clone truly is expanding, by the time we look at their blood in July, we would see that almost all of their monocytes now are mutant. There's been this large expansion. But what about a different possibility? What if this patient recently had a bacterial infection? Let's say the week before they came for their follow-up appointment, they got strep throat, which is a bacterial infection that we know changes cell types and their counts. And so this infection actually has changed the blood cell composition for this patient. And so what we're observing is actually an expansion of the monocyte fraction itself. And so while we are seeing more mutant monocytes, this is not a larger fraction of the monocytes than it was in January. And while we do have more of those monocytes, after this patient recovers from their bacterial infection, that monocyte fraction is going to shrink back down, and our VAF is going to be, again, 0.1. And so for this second possibility, we would actually not be concerned about clonal expansion. So I think from considering these two possibilities, it becomes clear that knowing cell type proportions really is critical for accurately interpreting changes in variant allele fraction. So for the remainder of my talk, I'd like to focus just on two questions. The first being, how can we quantify cell type proportions? And the second being, how does knowing cell type proportions change our interpretation of clonal hematopoiesis? So coming from a clinical perspective, what are some ways that we can capture cell type proportions? Well, it might seem obvious. Complete blood counts are something that are routinely captured for patients. We can just look from the CBC and see the proportions of the cell types that we have. But something to consider is that CBCs are performed on whole blood. And by the time you get to sequencing, through the processing and pre-processing steps, you actually lose some of the cell types that are available in the CBC. Specifically, neutrophils are a cell type that are highly sensitive to degradation and loss. And so by the time you get to the sequencing stage, the cell type proportions that you're looking at and that you're sequencing is actually different than what you would capture in a CBC. And so those two populations don't completely align. But an alternative that I'm very excited about is using DNA methylation to look into cell type proportions. So DNA methylation patterns are unique for cell types. And something that's really exciting in blood cell types is that each of those subtypes has a unique methylation pattern. And so this is something that we can use to predict cell type proportions. So if we look at a blood sample from a patient, you can quantify just all of the different times that you recognize a pattern for a lymphocyte, a pattern for a granulocyte, a pattern for a monocyte. And using just those counts, you're able to then directly infer the proportions of the cell types that you have in your sample. This is a method that was pioneered about 5 years ago by a number of different groups, and there are now a number of established methods for doing this. And the method that I employed is called MEthylCC. So this brings me to my hypothesis that quantifying cell type proportions with DNA methylation directly from those cells that we're going to be using for sequencing to determine the VAF will really improve interpretation of clonal hematopoiesis. So to evaluate this, I used a cohort of 34 patients recruited at Vanderbilt University Medical Center. These patients had been evaluated for clonal hematopoiesis and had given samples between December of 2020 and October of 2022, so about a 2-year period. These samples were taken at multiple time points from these patients, typically 2 to 12 months apart. So my first question with this data set was, well, how well do my methylation predicted cell fractions correlate with what we see in a CBC? So here are these plots I'm showing you for those 3 cell type groups. On the x-axis, you can see the CBC proportions, and on the y-axis, the methylation predicted proportions. And so the gray line going down the center would be if they were exactly the same. And so I think what you can appreciate from these R-squared values that we're seeing is that there's really high correlation actually between the CBCs and the monocytes, but depending on the cell type, you see kind of a shift away from that perfect line, and actually exactly in the way that we would predict. I'll draw your attention just to that middle plot of the granulocytes. So granulocytes is a larger bucket that encompasses the neutrophils, which I mentioned are the ones that are frequently lost. And you can see that consistently, the CBCs predict a higher proportion for the granulocytes than the methylation does. So directly confirming our hypothesis that these cell types that are captured are correlated, but actually not identical. And the methylation is aligned with the sequencing. So next, I wanted to see what does this mean for VAF. So I performed some linear modeling using cell type proportions and initial VAF. And so basically, I wanted to see what I could predict about a patient's next VAF given their initial VAF, given their monocyte fraction, given their granulocyte fraction, and given their lymphocyte fraction. And so I was able to predict linear models that could very highly predict a patient's next VAF just based on what I know about their initial and about the change in their cell type fractions. And so we see that this linear modeling actually explains essentially all of the variance in the VAF. And those dots are not very far off the correlation line, showing that for this set of patients, their variation has little to do with an expansion of a clone and more really to do with fluctuations in their cell type proportions. So what does this mean for classification of risk? Well, the way that risk is kind of described, evaluated in clonal hematopoiesis is by classification of clone behavior. Clones can be classified as expanding, stagnant, or shrinking, with expanding being the most concerning. And so I calculated clone behavior for these patients just based on their VAF. And doing that, I had a distribution of risks. And then I recalculated their classification using a cell type informed metric. And I saw a lot of change between categories. And actually a number of clones that seemed to be stagnant were now classified as expanding or shrinking. And similarly, clones that previously seemed to be expanding or shrinking with that greater setting and understanding now seem to be stagnant. And I think this has important implications for understanding which patients we actually need to follow up with versus which ones are okay. So in conclusion, I believe that it's really important to know cell type proportions. Knowing cell type proportions is critical for accurate interpretation of changes in VAF. From this work, we see that DNA methylation accurately quantifies cell type proportions for sequencing assays, and also that knowing these cell types proportions helps us to reclassify clone behavior and understand who is at risk. So to finish, I'd just like to acknowledge all of the people who have been involved in this work. My PI, Alex Bick, and Angela Jones, who runs the sequencing core at Vanderbilt and has worked closely with us on developing this assay, as well as several members of the Bick lab who have contributed to this work in important ways, and also my funding from NIH and from the CGC that has allowed me to be here. So thank you.